having a baby. Not now, of course, but in the future. But I'm a planner, and I don't really like to be unprepared. So we're gonna plan my future baby together. So what color hair do you think you should have? Actually, first we need a name. We can't start a baby without a name. Can I have a name?
Oscar.
Oscar. All right. Now, let's think about hair color. What kind of hair can Oscar have?
Red.
Red hair. Next, eye color. What kind of eyes should he have? What color? Cyan. Okay. I'm thinking that Oscar should be smart. We want to make sure he can make his way through this world, right? So I'm going to draw some glasses to show that he's very smart. He has a large head already, so I don't really have to add to that. We want to make sure Oscar is fit. So we'll draw him some muscles. And lastly, let's make Oscar immune to some diseases. Let's really make sure that he's a healthy young boy. So I was thinking we should put in cancer. Anyone else have any ideas?
Heart disease.
Heart disease, we'll write that one down. And lastly, let's write down cystic fibrosis, which is a genetic disease. Thank you for helping me plan my future son. Wouldn't it be so great if we could plan all of our children to make sure that they're all happy and healthy? But I mean, to do this, we need major technological developments. And no technological developments, as we've seen in history, have come without consequences. So August 6th, 1945, the first atomic bomb was dropped on Hiroshima, Japan, causing thousands of deaths and devastating destruction. Once the knowledge on how to create such a weapon was released, it could never be taken back, and now the reality is that 9 nations worldwide still harbor nuclear weapons as part of their self-defense. This acquisition of knowledge released a Pandora's box filled with massacre and endless paranoia. Recently, scientists have discovered a new tool that could change humanity's future, but for the worse or for the better. One thing we know for sure is that once this tool is used to edit our own genes, the world will never be the same. Let's just hope the result of nuclear weapons doesn't foreshadow the result of genetic engineering in human beings. So, hello everyone, my name is Elaine Thutt. I am a 4th year Flex student, and I'm here to talk to you today about progression and how not all progression should be embraced wholeheartedly without thinking about possible things that could go wrong as a result of this progression. So, to reach a form of compromise in which some of the benefits of genetic engineering can be enjoyed, while most of the risks are avoided, only certain areas of genetic engineering in human beings should be explored and tested until they can safely be applied to modern-day science, while other areas should be banned entirely to ensure that humanity doesn't become too wrapped up in controlling its own evolution. And if you don't understand this now, don't worry. I'm going to explain it all. So first, in this presentation, we're going to have a brief science lesson on what exactly genetic engineering is and what are the different categories you need to know. After that, we're going to talk about recent advances of this scientific field and what realistically this new tool can do as of right now. After that, we're going to talk about the hype surrounding the tool, why scientists are so excited about opportunities that this could pose. After that, we're going to talk about the moral controversies surrounding it. And lastly, we're going to talk about possible alternatives that you can have to using this technology in case you are morally opposed. So, before we launch into a moral debate on the topic, let's quickly explain what exactly genetic engineering is. So genetic engineering can be defined as the editing of an organism's genes to manipulate certain traits. And you can see an example of this every time you walk into your local grocery store. You go to the produce section and you pick out a super large, super ripe vibrantly red tomato. And I can almost assure you that no tomato naturally grows that way. A scientist somewhere has edited the genes of the tomato plant to really emphasize the traits that make it appealing to consumers. So that ruby red color, that huge size, they are products of genetic engineering. So the two different categories within genetic engineering are somatic cell gene therapy and germline cell gene therapy. And this just means that there are two different types of cells you can take DNA from to edit or change. The first being somatic cells. So somatic cells are any cells within your body that are not reproductive cells. And reproductive cells, as you all know, are eggs and sperm. And what it means to edit the DNA of a somatic cell is that the change only affects the individual the change is made in. Oftentimes, the changes that are made in somatic cells are temporary because the cell lifespan is so short, and it offers a therapeutic effect for diseases such as cancer or cystic fibrosis, etc. Germline cell gene therapy deals with germline cells, and germline cells are the flip side of somatic cells. They're reproductive cells, eggs and sperm. And if you edit the DNA of these cells, the changes are passed down to your entire progeny, meaning all of your descendants will carry the changes you make. When hearing about genetic engineering, you may come across the term genetic enhancement. And as the purpose of this presentation, we need to differentiate the two because they are two different things. Genetic enhancement is taking the genetic engineering above and beyond and using it for things that are not necessarily medical purposes. It's this idea of improving things, traits such as strength, height, intelligence, appearance, etc. Oftentimes, because of genetic enhancement, genetic engineering will be referred to as a slippery slope because people will think it will start off with good intentions by curing genetic diseases or making connections to common diseases in certain genes. And then it will eventually become an excessive use unnecessarily. So for things like changing your baby's eye color or changing your baby's hair color. And the reason why this is bad is because we don't want to become dependent on something that maybe we don't necessarily have to. So anyways, why are we talking about this now? Because to me, editing human genes sounds like some future dystopian novel, science fiction novel that is thousands of years in the future. And really the reason is because a new tool has been discovered called CRISPR-Cas9, and this tool can cut and paste DNA more efficiently and more accurately than ever before. So we had ways of doing this before, but CRISPR-Cas9 is kind of like the icing on the cake. It is supreme. So here is a video of Jennifer Doudna to kind of explain how CRISPR works and where it comes from. And she is a professor of molecular and cell biology at UC Berkeley, as well as chemistry, and she actually found and discovered this technology.
Showed they could use CRISPR to remove the DNA of a few years ago with my colleague Emmanuel Charpentier. I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to diffuse the bomb before it gets destroyed. So many bacteria have in their cells viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos.
All right, we're going to cut it off there. And just to give you a quick overview of what she was saying, CRISPR-Cas9 comes from bacteria. It's part of their self-defense system against viruses. And we wanted to take this tool from bacteria and use it for our own purposes within our own DNA. So, if you didn't quite catch it, she said at the very end, the Chinese scientists have already started to test human embryos with this tool. On August 18th of last year, they published their study, and after the study was published, a debate was sparked from the public. Many people thought that an ethical boundary had already been crossed, but we'll get into that more later. What they really found is that CRISPR-Cas9 is not ready for the advanced measures that we want to use it for. So, I'm sorry if I got you really excited about planning your future children, but the reality is CRISPR-Cas9 is not ready for use in human beings at this moment. When tested in complex organisms, it tends to have unpredictable results. This specific experiment was trying to remove a fatal blood disorder from 86 different embryos. Out of that, only 28 had the gene removed correctly, and even fewer than that had a new healthy gene inserted. So we see that this kind of has a high risk, low reward as of this moment. Oh, sorry. Eric Lander, a geneticist and professor of biology at MIT, states wisely in his article, The Brave New Genome, that it has been only a decade since we first read the human genome. We should exercise great caution before we begin to rewrite it. This quote really shows how fast-moving and fast-paced the science field surrounding DNA is. And because of this, only a decade ago we were reading what our DNA was for the first time, and now we want to rewrite it just 10 years later. Because we've only read it a decade ago, we can hardly understand DNA as much as we would like to. And it's really hard to edit something you don't fully understand. A hashtag was even made called #CRISPRFacts, and it's kind of a way to jokingly berate the people who get morally opposed to this topic, because in reality, CRISPR really isn't ready for any of the things that they foresee it doing. So let's talk about the hype. Why is there so much hype surrounding it, and why are scientists so excited about the possible opportunities?
Yep.
The reason that scientists are even exploring this field is because this technology could be a possible cure for genetic diseases. Genetic diseases usually occur because of a defect in just one gene, like sickle cell anemia is an example of this, cystic fibrosis, or Huntington's disease. And if CRISPR-Cas9 could be used to just take this one gene out and replace it with a new healthy one, we could have a cure for all genetic diseases, and if we use germline gene therapy, we could remove it from humanity's gene pool entirely. This is a pretty big deal. Another exciting aspect of this is that CRISPR-Cas9 could be used to form connections between more common diseases and different genes. So diseases pertaining to heart problems or diseases like cancer or Alzheimer's are very complex, and connections were being made before were being made through a slow process of trial and error in animals. With CRISPR-Cas9, we can speed this process up, but just a disclaimer, CRISPR-Cas9 does not necessarily provide a cure for these because they are so complex. They could contain roots in hundreds of different genes, and that is really quite hard to fix. And lastly, another reason people are very excited about this technology is kind of an extension of genetic enhancement. And that's the idea that if we could create a human race that could think quicker, run faster, climb higher, and feel deeper than ever before, stretching beyond our biological limitations as they are now to become improved in almost every sense of the world, why wouldn't we jump at that opportunity? We could be reaching humanity's full potential and alleviating possible unnecessary human suffering, according to Nick Bostrom, a philosopher at Oxford University, a professor in philosophy at Oxford University. So let's move on to the moral controversy surrounding the topic. And as you can see, I have a T-chart with the moral objections and the moral support. So we're going to go through the moral objections first. Many people are opposed to using this. Mostly these are concerning germline gene therapy, because CRISPR-Cas9 provides humanity with a lot of power. We would be now using this, picking the traits that move on and the traits that fade out. We are essentially shaping our own evolution with this technology. And who's to say that we can really know what's best? It's kind of giving us a God complex, as it says it right there. Another moral objection has to do with the loss of the individual. So, presumably if genetic enhancement ever became a reality, parents would be picking not only the physical traits of their future children, but also the personality traits. And a large part of the human experience, I believe, includes self-discovery, finding oneself. If parents are picking how their child is going to turn out, this is kind of taken away. And you're steered toward one path in life. You're losing your individuality. Another objection is loss of freedom, and that kind of goes hand in hand with the one previously stated. Even though genetics is up to chance as of right now, with genetic enhancement, parents are choosing specific traits, steering their children towards one path in life, and that kind of takes away with the child's freedom of choice as to where they want to go. And last is loss of equality. So presumably, again, if genetic engineering ever became a reality, it would not be accessible to everyone. It would probably be an expensive procedure that would need to be done, and then a distinction would be made between the genetically enhanced, who have an advantage, and the natural-born people. Moral supports for CRISPR-Cas9 and editing germline cells. One comes from an Oxford philosopher named Julian Savulescu, and he says, if we have the technology and know that it's safe, we are morally obligated to make our children the best that they can be. So if we know that we can make my future son, Oscar, happy and healthy, why wouldn't we want that why wouldn't we strive to give that to them? Another moral support has to do with the Hippocratic Oath that doctors take when they go into their profession. And it has to do with if this technology, we know it is safe, they, according to their oath, have to uphold that they will do anything they can to make sure that the patient becomes healthy again. And if that includes genetic engineering, then it is immoral not to use it. So, possible alternatives to this technology, if you are morally opposed after this presentation, are the things listed on the screen. And this is mostly for parents who may carry a recessive gene for genetic disease, and they want to have a biological child that is healthy. In different ways they can do this is PGD screening. This would mean taking several samples of the woman's egg and several samples of the man's sperm and fertilizing them outside of the body. Then they would screen each one for possibilities of genetic disease. The one least likely would be implanted into the mother, and then the natural order of the birth would go on. The next alternative is third-party reproduction, and this means that you are picking you're having a third party as part of your baby. So you would get a sample from a sperm bank or an egg donor. And by doing this, you are able to pick half of the DNA going into your child. And lastly is not a biological option for a child, but it's adoption, and it's always a viable option, and it should always be considered. So, to apply all my knowledge that I've learned this year through extensive research, I decided to write an editorial on why we should ban germline gene therapy. And I've submitted it to the Highlander, which is our school newspaper, and we'll be submitting it to the Birmingham Eccentrics student section so that I can really advocate for my opinion. I want you all to remember that editing germline cells means that changes are passed down to all of your descendants. No mistakes can be made because it is permanent. Although the rewards of gene editing within germline cells is tempting because we want our children to be happy and healthy, it's really not worth the risks because CRISPR-Cas9 holds so much power, and if something goes wrong, then we really can't change it. So, to be safe, I think that we should explore more into somatic cell gene therapy, which is a safer, more conservative option, rather than risking it with germline gene therapy. So, today we talked about somatic cells versus germline cells and the different therapies included in both of those, and what changing the DNA in each means. After that, we talked about improvements in the DNA field as of this moment, and what the technology can realistically do. After that, we talked about the moral controversy surrounding the tool, and lastly, we talked about different alternatives for parents who want a genetically healthy baby. So, it is well known that in World War II, Hitler dabbled with eugenics, entertaining this idea of creating a master race. Imagine if a tool like this fell into his hands. CRISPR-Cas9 holds a great deal of power, and it holds the power to change the human species. It only takes one person abusing this power to destroy our humanity. How much are you willing to risk? Thank you so much for coming to my presentation. I really hope you enjoyed it.